Hello boys and girls, this is Mr. So. Welcome back for another lesson of biology. Today we are going through lesson 26 of chapter 27, protein synthesis, the second part, translation. Before you start watching the video, remember to like and subscribe my channel and also share the video to your classmates. Let's start. Translation is a process of joining amino acid together to form a long chain polypeptide. And this process occurs at the ribosome. Did you remember what is ribosome? Ribosome, it is actually made up of something called the ribosomal RNA and protein, and it is a ball shape, as you could remember. And this is the site for protein synthesis, for you to join the amino acid to form polypeptide. And the diagram shown below is a electron micrograph. You can see this is actually the rough endoplasmic reticulum. It is called rough because on top of it you can see a lot of black dots, and these black dots, they are actually the ribosome. Ribosome, which are formed from the rRNA, the ribosomal RNA and protein, it can be found in different positions inside the cells. It can be found on top of the rough endoplasmic reticulum, the rough ER, or they can just be free floating on the cytoplasm. And their positions affects their production of protein as well. After the production of messenger RNA, mRNA in the nucleus, the mRNA carrying the codon is going to move to the cytoplasm and joined with the ribosome, with the start codon joining with the ribosomes first as the starting point. To allow amino acid to join with one another, you need a carrier of the amino acid. This carrier we call the transfer RNA, or in short, we call it the tRNA. This is the tRNA. On the top of the tRNA, it carries the amino acid. And at the bottom of the tRNA, it consists of the anticodon, which this anticodon is going to match with the codon on the mRNA. The tRNA carrying the amino acid will go into the ribosome and complementary base pair with the mRNA. Now the question is, there are anticodons at the bottom of the tRNA. Can you tell me what are the anticodons which match with the start codon on the mRNA? It should be A matches with U. U matches with A, and G matches with C. After joining of the first amino acid with the tRNA to the start codon, you need to join the second amino acid with another tRNA to the mRNA. For the second tRNA to join with the second codon on the mRNA, can you please tell me what will be the anticodon on the tRNA to complementarily match with the codon on mRNA? For C on the mRNA, it is going to match with the G in the anticodon of tRNA. A matches with U, G matches with C. Now you get the anticodon on the second tRNA. And after that, after there are two amino acids going together in the ribosomes, the amino acid, they will join together to form a bonding. After the joining of the first and second amino acid, in between the first and second amino acid, a peptide bond is formed. And the ribosome will move away from the start codon, and the first tRNA is released. After the release of the first tRNA and the ribosomes move away from the start codon, there are space opened up in the ribosome. This allows the third tRNA to bind with the mRNA, which carries the third amino acid. Now the question is, what is the anticodon on the third tRNA? Again, use the complementary base pair idea. C on the mRNA should pair up with G on the tRNA. And G should pair with C, A pair up with U. So now the anticodon on the third tRNA should be G, C, and U. And of course, a peptide bond is formed between the second and the third amino acid. The translation process, that means the joining of the amino acid, will continue when the ribosomes move away from the start codon, and it is going to stop when it reaches the stop codon on the mRNA. Why would the joining of the amino acid, the translation, stop? This is because for the stop codon, it does not code for any amino acid. This stop codon does not complementarily matches with any tRNA. There is no tRNA which carries any amino acid to join with the UAG codon. So we call this stop codon. Now, if there is no amino acid joining with the stop codon, this is going to be the ending site of the translation. After the ribosome reaching the stop codon, the polypeptide will be released. Translation occurs in the ribosomes. The tRNA carrying the first amino acid will join with the mRNA in a complementary base pair manner. And then the second tRNA will go in carrying the second amino acid. The first and second amino acid will join with each other by a peptide bond. The ribosomes will move away from the start codon. More joining of the amino acids to form a polypeptide chain. When the ribosome reaches the stop codon, the polypeptide chain will be released. The electron micrograph below shows you the process of translation inside a cell. The red one is the mRNA, the blue one is the ribosomes, and the green line, they are the polypeptide produced in the translation. Many ribosomes will move along the same mRNA at the same time to produce a lot of long-chain polypeptide. After the translation polypeptide formed, they will coil up and fold it up to form a protein, ball-shaped protein. Other examples such as one protein can join with another protein to form an even bigger protein. For the protein that are made at the ribosomes on the rough ER, they are usually the protein which are rather big, so they will be secreted as the digestive enzyme outside the cell, or they will be embedded on the cell membrane such as the carrier protein. They are rather big in size. For the protein produced by the free-floating ribosomes, the protein, they are rather small and they will remain in the cytoplasm and used by the cell itself, such as some internal use enzymes. Let's do a question to check your understanding. The sequence of amino acid in a certain part of polypeptide is shown below, with a lysine, proline, histidine, aspartic acid. So they are different amino acids. Don't worry, you don't need to remember their name and spelling. Don't worry. Okay, and which of the following is the correct sequence of the nucleotide on the non-template DNA? That means it's asking you the coding strand of the DNA. For this part, how can you do these questions? You have to first write out the mRNA sequence for these polypeptide. Let's do it together. Draw a straight line. This is the mRNA. And on top of it, let's write down the codon for different amino acid. For lysine, for example, lysine, it is having a codon AAG. Let's write down AAG. And then for proline, it has the codon of C, U. And then for histidine, it has a codon of C, A, U. And the last one, aspartic acid, it has a codon of G, A, U. Alright, so now you get the mRNA sequence. How do you get the coding strand DNA for this question? That is, you have to do a little drawing. Draw out the DNA into two separate strands, with the lower strand as the template, and the upper strand as the coding strand, and the middle part, you have the mRNA. And now, what do you know about the mRNA? That is, it has the AAG sequence, and then CCU codon. Okay, write out. What do you know about the mRNA and the coding strand? That is, the mRNA and coding strand, they're exactly the same, except the base U in the mRNA should be changed to base T in the coding strand. So therefore, when you change it to a coding strand, what is the coding for the coding strand? It should be A, G, makes sense, and then C, no more U, sorry. The U is now changed to T. And then C, A, no more U, it should be T, change it to T. And then G, A, no U, change it to T. So this is the coding strand. Now what is your answer? You should get your answer as A. Okay, separate into the codons, 3 in a group, separated into a triplet code, 3 in a group. So you have the exact answer as my drawing. That's it about a part of protein synthesis part 2 translation. Please go to my next video to do the classwork for the protein synthesis. Classwork 27.1. I will see you in the next video. Bye bye.